Hi guys, we're going to talk about photosynthesis today, and boy oh boy is it going to be fun. So you already know some of this stuff, but let's just get the basics down. Photosynthesis is shown by this reaction. You have 6 carbon dioxide molecules, 6 water molecules. Sunlight is going to come in. It's not really a reactant, but I have it listed as a reactant here. And you're going to end up with all of that is going to come together to give you just 1 glucose molecule and 6 oxygen molecules. So again, 6 CO2 plus 6 water, and then sunlight comes in, and you're going to get 1 glucose molecule and 6 oxygen molecules. If you don't know anything else about photosynthesis by the time we're done with this, you need to know that. Remember that photosynthesis happens in the chloroplasts, and what it does is it converts the sun's energy into stored chemical energy in the form of glucose. Chloroplasts, remember, are made of a double membrane, and then even inside that inner membrane is a membrane called the thylakoid. And that thylakoid stacks up into these little stacks called the grana. So remember before I told you that they were like pancakes stacked up on top of each other? Well, we don't get to call them coins or pancakes or anything anymore. We have to call them grana. That's their appropriate name. And then the specific membrane that's making up the grana is called the thylakoid. Then there's space between outside the thylakoid membrane, and that's called the stroma. So if you look in here, you have your outer membrane, you have your inner membrane, and then even inside the inner membrane, there's another membrane called the thylakoid. And the thylakoid membrane stacks up into these grana. So one of them is a granum, one stack is a granum, but the whole group of them is called grana. All right, so again, outer membrane, inner membrane, and inner membrane. Then you have the thylakoid membrane that's stacked up into these grana. And these grana are important for one part of photosynthesis. And then all of this space out here is called the stroma. And the stroma is going to be important for another part of photosynthesis. So there are 2 sets of reactions that are going to come together to make sure that photosynthesis can happen. We have light-dependent reactions and light-independent reactions. Now, just looking at those names, I bet you can figure some cool stuff out. I bet you can figure out that that first set of reactions requires light. That second set of reactions doesn't require light. So let's talk about light. So on Earth, sunlight appears as white light. So if you look at bright light, you think of it as looking white. White light actually contains all the different colors of the light spectrum. So you guys have probably seen a prism, or at least a picture of a prism at some point before. So if we send in white light into the prism will break apart that light into all of its component colors. And that gives us our good Roy G. Biv rainbow of colors here. Okay, so that white light has all of these other colors inside of it. We just can't see it most of the time. Well, autotrophs can see that light because they have pigments. Pigments are just little molecules that absorb light. Pigments are little molecules that absorb light, but depending on what pigment they are, they're going to absorb different lights. So chlorophyll A is going to absorb red and blue light. Chlorophyll B can absorb blue and orange light. Well, what's not getting absorbed here? That's right, green and yellow. Green and yellow light are not getting captured. So if green and yellow light are not getting captured, what does that mean is happening to them? Well, they're being reflected. Now, it might seem weird to you that green plants aren't absorbing green light, except that's exactly why they're green. We see them as green because they absorb all the rest of these colors of light, and then the green and the yellow is what gets bounced back at us. And if that's what gets bounced back at us, that's what our eyeball actually sees. So all of these colors go in, but the green and yellow bounces off and doesn't get absorbed. So the chlorophyll is going to absorb all of the red and all of the blue and all of the orange. That's all going to get absorbed, but the green and yellow get bounced back. Okay, so the light-dependent reactions, light is required. You have to have light for the light-dependent reactions to happen. If you were to keep an autotrophic plant in a closet, I don't know what other kind of plant it would be if it weren't autotrophic. If you were going to keep a plant in a closet completely dark and devoid of light, it would die very, very quickly because the light-dependent reactions can't happen. The light-dependent reactions happen in the grana, and remember that the grana is made up of membrane. So that thylakoid membrane is what makes up the grana. So the thylakoid is really where those reactions are happening, that membrane. And remember, the more increased surface area we have for that membrane, the more reactions we can have take place. That's why that grana is stacked like that. Okay, so if we look at our whole chloroplast here, our light-dependent reactions are going to happen right here in the thylakoid of the grana. Don't worry about all that other mess on that slide. Okay, so let's zoom in and look at the thylakoid membrane. There's a lot going on in here, but don't worry, I'm going to walk you through it. So the light-dependent reactions start when light hits photosystem 2. This guy is photosystem 2. This big green glob is photosystem 2. See these little tiny green guys? Those are electrons. So photosystem 2 is basically just a bunch of chlorophyll molecules and a bunch of proteins, enzymes, that are hooked up in there, creating this little glob of awesomeness that's going to work together. So light's going to come in and hit photosystem 2. And when light hits photosystem 2, those electrons are going to get really excited. So keep an eye on those electrons. And it gets so excited, photosystem 2 gets so excited by the light that those electrons just bust loose, okay? And when those electrons bust loose, they're super high energy. They're super excited, they've got lots of energy, okay? At the same time that that's happening, light is gonna hit water, and that water is gonna break apart. So take a minute and think about what happens when water breaks apart. What do you know happens when water breaks apart? Yes, sort of. So light's going to hit this, and what we're going to get in this case, rather than a hydroxide ion, which is what you're thinking probably, I hope, rather than a hydroxide ion, the whole entire thing is going to come apart. And as the whole entire thing comes apart, you're going to end up with 2 H+ ions. And remember, an H+ ion is really just a proton, because remember, a hydrogen ion or a hydrogen atom by itself is a proton and an electron. Well, we're going to bust the electron loose. And so it's really just a proton then. So whenever you see H+, try to keep in your head that that's really just a proton, okay? So we've got 2 protons, we've got 2 electrons, and then we've got our oxygen, or we've got our oxygen atom, okay? If we do that twice, so if we bust 2 of those apart, our light comes in and hits, we get busted out, but watch what happens with our oxygens, because remember, our oxygens like to be friends with each other. So if that happens with 2 water molecules, you get 2 H+ ions, which are really just protons, you get 4 loose electrons, and you get a diatomic oxygen molecule. Well, conveniently enough, we know that one of the byproducts of photosynthesis is diatomic oxygen. This is where it comes from. This is where that diatomic oxygen comes from. Right away in the first step of photosynthesis, we bust that water apart, and when that water gets busted apart 2 times, those oxygens come together, form O2, that leaves. Okay, so why did we need to bust that water apart? Well, remember that the light came in and hit photosystem 2. The light came in and hit the water at the same time. Those electrons in photosystem 2 got really excited and they popped out. Conveniently, we just created more excited electrons, and those are going to go into photosystem 2 to replace the ones that just got released. At the same time, the O2 is going to get released. Okay, so now those excited electrons that left photosystem 2 are going to go bouncing down the membrane, okay? But as they go bouncing down the membrane, they're going to lose energy, because when you bounce a lot, you get tired, and that happens to the electrons too. So they get carried down the membrane and they lose energy. Luckily enough for them, guess what's going to happen? More light is going to come in. Light is going to hit them while they're in photosystem 1. So light hits them again. Make a prediction. What's going to happen when light hits them? Good. They're going to get excited. And when they get excited, what's going to happen? Good. They're going to pop out. Okay? So they get so excited and they fly out again. This time, though, they're not going to go bouncing down the membrane. This time, they're going to get grabbed onto by these special electron-carrying molecules called NADP+. Okay? Now, in your head, think about the fact that there's a plus there. And what we want them to grab is an electron. Okay? So we've got the positive of the plus, and we want them to grab an electron. Okay? So that should make sense to you then, that they're going to grab electrons. What they're actually going to do, though, is they're going to grab 2 electrons. Eek. Okay, so NADP+ and 2 electrons Now we're overly negative, right? Eek. But what we're going to do is, if you think of NADP plus as being like a jar, and you've got super excited electrons all over the place, you've got your jar of NADP plus, you're going to put 2 electrons into your jar, and then you need a lid. You want to guess what the lid is? I don't care if you want to or not. Guess what the lid is. What's the lid going to be? Hopefully you said a proton, which is really just a hydrogen ion. So we had NADP plus, and then we're going to bring in H plus. So that's a plus and a plus. And those are going to balance out the negative and the negative that are going to jump out, okay? So you've got the NADP plus that's going to scoop up the 2 electrons in the jar, and then the hydrogen is like a lid that goes on top, okay? That's an analogy that you need to remember because it's going to become important later on, okay? So NADP plus comes in, scoops up 2 electrons, a lid goes on. Another NADP plus comes in, scoops up 2 electrons, a lid goes on, okay? So NADP plus comes in, grabs the electrons, puts the lid on, becomes NADPH, and then those guys are going to go off to the next part of photosynthesis. At the same time all of this is happening, you've got protein pumps that are in the membrane. These guys, these purple guys are protein pumps. And what they're doing is they're pushing hydrogen ions inside the membrane. But look at our concentration gradient. We have a lot of hydrogen ions or protons inside the membrane already. We have very few outside. And so if we want our protons to go inside from outside, if we want our protons to go this way, we need a protein pump to do that because we're going against the gradient. So we're going to pump those guys in there. And as they do that, you're going to have a huge positive charge over here and a huge negative charge over here. And that's going to stress out ATP synthase. ATP synthase, look, that ends in ACE. What does that mean it is? Good, it's an enzyme. ATP synthase is an enzyme, and it's going to get so stressed out, it's going to spin. Okay, so it's going to spin around and spin around and spin around. As it spins around with those protons going through it, ADP, remember adenosine diphosphate, is going to grab a phosphate and get turned into ATP. Let's watch that again. ADP becomes ATP. So that third phosphate gets added on there because ATP synthase is spinning, okay? As that happens then, the ATP is going to move off to another reaction, okay? So, the light-dependent reactions. The light comes in and excites the electrons in photosystem 2. The light breaks apart the water. The electrons from the water go into photosystem 2 to replace the ones that just popped out. The oxygen atoms from the water are going to find each other and form diatomic oxygen, which gets released. That's one of our byproducts. The excited electrons are going to go bouncing down the membrane. The excited electrons are going to get captured by the NADP plus, and NADPH is going to get formed. The protons are getting pumped across the membrane, and you've got this gradient now. That's going to stress out the ATP synthase, which is going to spin, and that's going to cause ADP and ADP and a phosphate to come together to make ATP. Even more abbreviated, light hits, water's broken, oxygen formed, NADPH is formed, ATP is formed. That's the super abbreviated version. But those are just the light-dependent reactions. Now what? Well, now we have the light-independent reactions. The light-independent reactions are also called the Calvin cycle. Light is not required for the light-independent reactions. It doesn't have to be there, but it's not a problem either. So it's not like these have to happen in the dark. And in fact, back in the day when some of us were in school in our caves, they called these the dark reactions. And they called them the dark reactions because they don't require light, but it led a lot of people to think that they had to happen in the dark. They don't have to happen in the dark. They can happen whenever they need to happen. So light-independent reactions don't require light, but light's not a problem either. They happen in the stroma. So again, let's go back to this diagram. Remember, our light-dependent reactions happen in the grana here in the thylakoid membrane, but now this stuff is going to happen in the stroma, so out in this empty space here. Well, it's not really empty, but okay, so already floating around in the stroma are these molecules called RuBP. Okay, they're not really called RuBP. It's ribulose bisphosphate. But ribulose bisphosphate is not very easy to say. It's also abbreviated like this, capital R, lowercase u, capital B, capital P. So I call it RuBP. Notice that RuBP are 5 carbon molecules, okay? We're going to use 3 of them, and we're only going to look at the carbon molecules, at that carbon skeleton or carbon backbone, because it is way too hard to look at the whole entire thing. That just gets way too confusing. So we're just going to look at the carbon skeleton. So these are 5 carbon molecules. They're already there, okay? 3 carbon dioxide molecules are going to come floating by. That's convenient. We've got 3 RuBPs. So our 3 RuBPs are going to meet up with our 3 carbon dioxides. But another enzyme called Rubisco, okay, this awesome enzyme called Rubisco is going to come in and help. And what Rubisco is going to do is it's going to help one RuBP combine with 1 carbon dioxide. And when that happens, you're going to get 2 molecules that each consist of 3 carbon chains. So if you look at it, you've got 6, 6, and 6. And then quickly it breaks apart into 3 and 3. But then remember that we did that 3 times. And so when we did that 3 times, that's why I have 6 molecules up here, okay? Don't worry about what happens to the oxygens. The oxygens go off and they make water and they do some other cool stuff and just blah, 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 blah. Don't worry about the oxygens. Just worry about the carbons right now, okay? So now we took 1 RuBP, which was 5. We added a carbon dioxide, which was 6, but then it broke apart into 2 3s, okay? So we've got these 3 carbon chains. Okay, sorry guys, I had to move. The bell rang and a class came in. So remember those NADPHs that we made in the light-dependent reactions? This is why we made them. They're going to come in and they're going to drop off their electrons. But in order to drop off their electrons, remember we have to take the lid off the jar. And so if we have to take the lid off the jar, those electrons are going to come off, but now the lid is off too. Okay, so we've got our NADP pluses back again. That's our jar. The electrons get dropped off, and then the proton, or the lid, that comes off too. Okay, so the NADPHs come in and they drop off their electrons. We take the lid off the jar, we've got the jar, we've got the lid, and the electrons come out. The electrons come out because we need them for these guys. Okay, that NADP plus is recyclable, and it's going to go back to the thylakoid, and it's going to go scoop up more electrons and put a lid on, and then come back and it's going to drop off more of these guys, and it's going to scoop up electrons and put a lid on, and then come back and drop them off, and NADP plus just goes back and forth and back and forth and back and forth and back and forth. Okay, those protons, they're really just going to hang out in the stroma, and they're going to do whatever protons need to be. They can be lids again if they need to be lids again. They can get pumped across the membrane for that concentration gradient so that the ATP synthase can spin. They can do whatever they need to do. Okay, and then remember that ATP, we made all that ATP also when the ATP synthase was spinning. That ATP is going to come in too, and it's going to drop off some energy. And when it drops off that energy, it's going to do the same thing. It's going to break apart into pieces. That energy is going to get used by our molecules. Those molecules are going to get converted again. And then the ATP can go back to the thylakoid to do whatever it needs to do. The ADP can go back to the thylakoid to do whatever it is that it needs to do. Okay? So now we've got these new improved 3 carbon molecules. One of those, only one of those, gets to go off and is eventually going to become a sugar, okay? For now, you can think of it as half a glucose molecule, okay? 3 more ATP are going to come in, and what those 3 ATP do is they come in and they're going to help those little 3 carbon molecules turn back into RuBP. How many carbons were in a RuBP? 5. Good, okay. So those ATPs are going to help RuBP form again. Those ATPs are going to go back, and the ADPs are going to go back, and they're going to get recycled. And now we're right back where we started, and the whole thing can happen again. So our 5 carbon molecule can attach to carbon dioxide, become a 3 carbon molecule, NADPH and ATP come in, drop off energy and electrons. A new one of those 3 carbon molecules gets made. Okay, we've got another half a glucose. Our RuBP gets regenerated again, and now our 2 half glucoses become a whole glucose, and now our whole glucose is a glucose. Yay, we did it. Awesome sauce. That was a lot of work for 1 sugar molecule. Okay, so we call this the Calvin cycle because it really is a cycle. So really quickly, this is how you'll see it made most often. So here are our 3 carbon dioxides. Here are our 5 RuBPs. Okay, we've very briefly got these 6 carbon molecules that are going to break apart into 3 molecules. So we've got 6 3 carbon molecules. Okay, and then our ATP comes in, our NADPH comes in, we drop off energy, we drop off electrons, we've still got our 3 carbon molecules. One of those goes off to become a glucose, the other 5 get recycled, more ATP comes in to help them get recycled, and they become RuBP again, and they go through again with 3 more carbon dioxide molecules, till we have another 3 carbon molecule, and now we've got an entire glucose made, and our RuBP needs to get recycled. Whew, that's a lot of work. Okay, so summing up the light-independent reactions are the Calvin cycle. Carbon dioxide comes in, meets up with RuBP that's already there. It's a 5-carbon molecule, and then Rubisco. Remember, Rubisco is that enzyme that's going to help that happen. 3 carbon molecules form. So the molecules that are made of 3 carbons, not 3 molecules. Okay, so each RuBP and a carbon dioxide is going to give us 2 of those molecules that are 3 carbons each. NADPH drops off electrons, ATP drops off energy, NADP plus and NADP go back to the thylakoid. Those 3 carbon molecules get converted back to RuBP. 3 turns of the cycle make a half a glucose, 6 turns of the cycle make a glucose. So carbon dioxide, carbon dioxide, carbon dioxide, carbon dioxide, carbon dioxide, carbon dioxide. Now you've got a glucose. So carbon dioxide, NADPH, and ATP come in. They react with RuBP to create glucose after 6 turns of the cycle. NADP plus and ADP and RuBP get recycled. All right, so that's it except for this question. There is no light involved in the light-independent reactions, but Gnant says you have to have light in order for them to happen. Gnant says you can't have the Calvin cycle without light. What? I just went through this whole big thing about how you don't need light to do the Calvin cycle, but I say you do need light to do the Calvin cycle. Why? See if you can figure it out. If you can't figure it out, well, okay, see if you can figure it out. Then pause. Think about it. Think about it. If you pause and you think about it for a few minutes and you still can't figure it out, start this back up again because I'm going to show you another picture that might help. So pause. Okay, if you're struggling, take a look at this picture. This picture might help you get to the answer. Why do you need light in order for the Calvin cycle to happen? If you still can't figure it out, ask me about it in class tomorrow. Bye.